related to all biology streams. Okay, let's start with the discussion. So the first question is, dash is the basic unit of life. ఈ క్రింది వాటిలో ఏది జీవం యొక్క ప్రాథమిక యూనిట్? There are four options given. First one is protein. So protein is a biomolecule and it is a polymer of amino acid, and protein involved in all types of contractions or movements in our body. So proteins are considered as running horses of living body, and proteins sometimes act as enzymes and involved in metabolic processes. So protein is not a basic unit of life. And second one is DNA. So DNA is a genetic material and it is a polymer of nucleotides, and it is involved in replication and transcription. So it is also not the basic unit of life. Third one is cell. So you all know cell is the basic unit of life or living forms. So when you are constructing a house, brick is the basic unit. Similarly, for any living organism, cell is the basic unit for living organism or life. So cell is the right answer. And fourth option is chromosome. Chromosome is a vehicle for inheritance. That means it harbor the DNA and carry from one generation to another generation. So chromosomes are the vehicles for inheritance, or vehicles for the DNA transfer from one generation to another generation. So for this, cell is the right answer. Remaining all are biomolecules, and chromosome is the vehicle for inheritance. The second one is, dash is the prominent difference between prokaryotes and eukaryotes. Prokaryote మరియు eukaryote కణాల మధ్య అతి ముఖ్యమైన వ్యత్యాసం. You all know prokaryotes means no nucleus, without nucleus. That means nuclear membrane is absent. Whereas eukaryotes means U means true, karyon means nucleus. There is a nucleus present in eukaryote. That means nuclear membrane is present. This is the prominent difference between prokaryotes and eukaryotes. If you see the options, the first one is nuclear membrane. Yes, it is the right answer, because prokaryotes doesn't have any nuclear membrane, whereas eukaryotes contain prominent nucleus and it's surrounded by nuclear membrane. The first one is right answer. If you take the other options, take second option, that is cell wall. Cell wall present in both prokaryotes and eukaryotes. For example, bacteria, it contains cell wall made up with peptidoglycan. Bacteria is a prokaryote. It contains cell wall made up with peptidoglycan. To take eukaryotic organisms, except animals, remaining all eukaryotes contain cell wall. For example, fungi. Fungus contains cell wall, which is made up with chitin. It is also fungus, also eukaryote. Similarly, plants. Plant cells contain cell wall made up with cellulose and hemicellulose and pectin. So plants also contain cell wall, except animals, remaining all eukaryotes contain cell wall. So cell wall is not a prominent feature of prokaryote and eukaryotes. Third one is ribosomes. Ribosomes present in both prokaryotes and eukaryotes also. Fourth option is none of them. So the correct answer is nuclear membrane. The prominent difference between prokaryotes and eukaryotes is nuclear membrane. Next question is, in prokaryotes, pro means without nucleus. That means bacteria. The ribosomes are, as I said earlier question, both prokaryotes and eukaryotes contain ribosomes. But the type of ribosomes present in prokaryote differs with eukaryotes. There are four options given. The type of ribosomes present in prokaryotes, four options given. First one is ATS option. So ATS ribosomes present in eukaryotes. Those organisms which contain nucleus, they have ribosomes of ATS. For example, if you take plant cells contain 80s ribosomes. Animal cells contain 80s ribosomes. Fungal cells contain 80s ribosomes. Protista cells also contain 80s ribosomes. All eukaryotic cells contain 80s ribosomes. Second option is 60s. 60s is not a complete ribosome. It is a large subunit of 80s ribosome. If you dissociate 80s ribosomes into two subunits, large subunit and small subunit, the large subunit of 80s ribosomes is 60s and the small subunit of 80s ribosomes is 40s. So second option is also wrong. Third option, 70s. This is the correct answer. All prokaryotes contain 70s ribosomes. And fourth option is 80s and 70s. If the question is given like this, that means in eukaryotes, the ribosomes present are, then the answer is 80s and 70s because in eukaryotic cells, cytoplasm contain 80s ribosomes. Eukaryotes, mitochondria and chloroplast is present. These mitochondria and chloroplast contain 70s ribosomes. If this question is given like this, that is, eukaryotes contain which type of ribosomes means both 80s and 70s. Here they are asked in prokaryotes. So prokaryotes contain only 70s ribosomes. So the option is 3. We take the next question. Due to activity, lysosomes are called as suicidal bags. Lysosomes are called lysosomes ni suicidal bags అని ఏ యాక్టివిటీ వలన అంటారు? So first ఇక్కడ 4 options given. First option is metabolic. So lysosome is a cell organelle. So in the cell organelle, no metabolic process takes. Generally metabolism occurs in cytoplasm or chloroplast or mitochondria only because they contain genetic material or genetic material supporting molecules. So metabolism occurs only in chloroplast, mitochondria, cytoplasm. Remaining all cell organelles, there is no metabolism. So metabolism means that is wrong. And second option is catalytic. So catalytic means enzymatic activity. Suicidal bags doesn't show any enzymatic activity because they contain some phosphatases, but because of that phosphatase activity only, lysosomes are not considered as a suicidal bags. And third one is parasitic activity. Parasite means foreign material. Any foreign material enter into body, then it is called parasite. But lysosomes are cell organelles only. So we should not call lysosomes as a parasites. And the fourth option is hydrolytic activity. This is the correct answer because whatever the aged organelles or aged proteins or biomolecules enter into lysosomes, the phosphatase activity of lysosomal enzymes degrades those aged molecules and recycle them. Because of that reason, lysosomes are considered as suicidal glands. So hydrolytic activity of lysosomes is mainly due to this activity only, lysosomes are considered as suicidal glands. And another thing is, lysosomes are considered as highly polymorphic organelles in the cell. What are the highly polymorphic organelles in the animal cell means lysosomes. So lysosomes are the highly polymorphic organelles in the animal cell. They are the 4 questions with explanation. Take another question. Which of the following is found with bulbi అనే rings? సో కింద వాటిలో ఏది బాల్బీ అనే రింగులతో కనబడుతుంది? We take the options. First one is polytene chromosomes. So polytene chromosomes contain bulbi rings. These polytene chromosomes generally found in salivary glands of insects. Salivary glands of insects. Why these chromosomes are abnormal means in these salivary glands of insects, repeated replications taking place, but chromosome separation not taking place. Because of that reason, repeated replications of DNA leads to large amount of DNA concentrated in particular nucleus. Because of that reason, they become fold for multiple times. They fold and form these ring-like structures called Balbiani rings. So polyethylene chromosome exhibits Balbiani rings, and it is observed in uh, salivary gland cells of insects. The second option is known as heterosomes. Heterosomes are nothing but allosomes. We take human cell. Human cell nucleus contain 46 chromosomes. In these 46 chromosomes, 44 chromosomes are autosomes. That means they are responsible for the somatic characters. And another two chromosomes are called allosomes or sex chromosomes or heterosomes. So these heterosomes or allosomes or sex chromosomes are responsible for the development of secondary sexual characters in the humans or animals. So heterosomes are nothing but allosomes. So heterosomes or allosomes or sex chromosomes doesn't contain any Balbian neurons. And autosomes, as I said earlier, every human cell nucleus contain 44 autosomes. So these 44 autosomes are responsible for the somatic characters. So these also doesn't contain any Balbiani. Another thing, lamp brush chromosomes. These lamp brush chromosomes found in oocytes of all animals, all types of animals. So these lamp brush chromosomes are present in oocytes of animals, mostly in lower animals like insects, arthropods, like that. Animals they are. So lamp brush chromosomes are highly extended chromosomes. That means there is no highly concentrated DNA. This is highly extended form of DNA is present. So they are like brush-like structures, but not, but they doesn't contain ball bearing rings. So which of the following you found in ball bearing rings means polyethylene chromosomes. If you take the difference between polyethylene and Lamb-Burr's chromosome, these are the two different abnormal chromosomes. If you take the differences, definition, polyethylene chromosome means a giant chromosome present in the cells of the salivary glands of the diphtheria fly species found in some diphtheria or insects. Lamp-breast chromosomes. These are also giant chromosomes present in growing oocytes of most animal species. Oocytes. They are present in oocytes. Polyethylene chromosomes present in salivary glands. Lamp-breast chromosomes present in oocytes, but both are giant chromosomes. And polyethylene chromosomes were discovered by Balbiani, whereas lamp-breast chromosomes discovered by Fleming. Next, third one, appearance. Six armed chromosomes with the DNA strands construct a dark and white hands. They contain six arms chromosomes. Generally, each chromosome contains two arms or four arms, but these polyethylene chromosomes contain six arms. And if you stay in that chromosome, it forms dark and white bands. Whereas Lambras appearance, they appear like a Lambras chromosomes appear like a brush-like structures. Presence. In salivary gland, cells of diphtheria fly species. So they are present in salivary glands. Whereas Lamprey's chromosomes present in oocytes of vertebrates. Size comparatively smaller than the Lamprey's chromosomes because the polyethylene chromosomes are smaller than Lamprey's chromosomes. The largest chromosomes are Lamprey's chromosomes. Lamprey's chromosomes are the largest chromosomes structure made up of many DNA strands. Polyethylene chromosomes are made up with many DNA strands, whereas Lamprey's chromosomes made up of main chromosomal axis with a lateral loops going on opposite directions vertical to tip. So it contains main strand as a chromosomal axis with lateral loops going on opposite direction vertical to it. So this is the main differences between polyethylene and Lamprey's chromosomes. So bulbinary rings present in polyethylene chromosomes. Next, another question. Cytokinesis is the term used for division of. Cytokinesis అనేది మనం ఎక్కడ ఉపయోగిస్తాము in general cell division contain karyokinesis and cytokinesis. Karyokinesis means nuclear division. Cytokinesis means cytoplasm division. So the answer is cytoplasm. Cytokinesis means cytoplasmic division. We take another option. First one is synapsis. Synapsis means in cell division. We have two types of cell divisions. One is mitosis and another one is meiosis. In meiosis, we have two subphases, meiosis 1 and meiosis 2. In meiosis 1, again, prophase 1, metaphase 1, anaphase 1, telophase 1 are present. In this prophase 1, there are homologous chromosomes come and contact together and form homologous pairing. This homologous pairing formation taking place, then the chromosomes attach each other. This process is known as synapsis. So, in prophase 1 of meiosis, the homologous chromosomes come and together and form bivalents or tetrads. This process is known as synapsis. So, synapsis means that. And second one is cell nucleus. Karyokinesis related to cell nuclear division, whereas cytokinesis is related to cytoplasmic division. So cell nuclear division means karyokinesis. Third option is cytoplasm. Cytokinesis means cytoplasmic division. Fourth one is chiasmata. Chiasmata means X-shaped structures formed during prophase 1 of meiosis 1 of meiosis. So these X-shaped structures formed between homologous chromes of non-sister chromatids are called chiasmata. In this chiasmata place, actual crossing over taking place. So chiasmata are responsible for the crossing. Chiasmata, how chiasmata taking place means non-sister chromatids of homologous chromosomes. The X-shaped structures formed between non-sister chromatids of homologous chromosomes is called chiasmata. The chiasmata is the place where crossing over taking place. So this is about other options of the questions. Another question. DNA is not present in. DNA అనేది ఏ ఆర్గనల్స్ లో లేకపోతే దేంట్లో ఉండదు. ఫస్ట్ వన్ ఈజ్ న్యూక్లియస్. న్యూక్లియస్ ఇస్ ద హార్బర్ ఆర్ ప్లేస్ ఫర్ డిఎన్ఏ. సో న్యూక్లియస్ ఈజ్ ఏ రాంగ్ ఆన్సర్ బికాస్ న్యూక్లియస్ కంటైన్ డిఎన్ఏ. And second one is lysosomes. Lysosomes are the cell organelles but doesn't contain any DNA material. So lysosomes is the right answer. Mitochondria also contain DNA. Mitochondria contain circular DNA. Number of copies of circular DNA is present. Chloroplast also contain DNA. So chloroplast also contain circular double-stranded DNA. So nucleus contain DNA. Mitochondria also contain DNA. Chloroplast also contain DNA. But lysosome doesn't contain any DNA. So if you take nucleus, nucleus contain linear DNA. Linear shaped DNA present. Whereas mitochondria and chloroplast, circular DNA is present. Circular DNA also number of copies present. That's why mitochondria and chloroplast are called semi-autonomous organelles. Mitochondria and chloroplast are considered as semi-autonomous organelles because they can make some of their metabolites within the mitochondria or chloroplast, and the remaining they get from nuclear DNA or cytoplasm. So that's why they are called semi-autonomous organelles. Mitochondria and chloroplast are semi-autonomous organelles, and according to some theories, mitochondria and chloroplasts are bacteria in nature, bacterial origin. So mitochondria and chloroplasts, according to endosymbiotic theory, mitochondria and chloroplasts are bacterial origin. Once upon a time, they are bacteria, and now they become part and parcel of the eukaryotic cell. So mitochondria, the chloroplasts are bacterial origin according to endosymbiotic theory. So which organelle doesn't contain DNA? That is lysosomes. The next question is alleles are యుగ్మ వికల్పకాలు అనేవి. These are the alternative forms of genes. If we take, we are all diploid organism. For example, human, we take each and every cell contain nucleus, and each nucleus contain 46 chromosomes. In these 46 chromosomes, 23 we are getting from father and 23 we are getting from mother. Father first chromosome, mother first chromosome. Similarly, father second chromosome, mother second chromosome. Similarly, father third chromosome and mother third chromosome are called homologous chromosomes. In each homologous chromosome, two copies of particular gene present. that two copies of particular gene are called alleles. So alleles means genes of alternative forms of gene copies are called alleles. We take first option, alternative forms of DNA. So it is not related to DNA, it is related to genes. So alleles related to genes. So first option is wrong. Second option is alternative forms of RNA. So it is not related to RNA, it is related to genes. Third option is alternative forms of chromosomes. No, alternative forms of chromosomes not related to chromosomes. As I said earlier, it is related to genes. So alleles means related to genes. Alleles are alternative forms of genes. Applied organisms, the number of alleles are 1. Diploid organism, the number of alleles are 2. Triploid organism, the number of alleles are 3. Tetraploid organism, the number of alleles are 4 particular gene. So the ploidy increases, the number of allelic copies also increases. So alleles means related to genes. Take the next question. The process of suppressing one gene activity by non-allelic gene activity is known as. Generally, every gene or every character is regulated by two forms. Those are called alleles. If same allelic genes or copies suppresses another gene, another copy, then it is called dominance. If another case, process of suppressing one gene activity by another gene, non-allelic gene activity, then it is called epistasis. For example, if you take hypostasis, hypostasis means suppressed gene is known as hypostasis. If anyone is suppressing, the suppressing gene is an epistatic gene. The suppressed gene is known as hypostasis. Hypostasis means the suppressing of a gene. Suppressing of a gene is known as hypostasis. So this is not the correct answer. Next, pseudodominance. Pseudodominance generally we observed in haploid organisms. What is pseudodominant? Pseudodominance means in haploid organism. Haploid organisms means each and every gene has only one copy, and that one copy express its character. If two copies are present, one gene is suppressed by another copy. So it is dominance. If one copy is present, one copy only present, that in haploid organisms are there. So there is no dominant nature. So the whatever the gene is present, that gene is expressed. So this is called pseudo dominance. Whenever dominant gene is absent or dominant gene not at all present or dominant gene inactive, then the suppressed or recessive gene shows its character. This is called pseudo dominance. Pseudo dominance means in haploid organism, whatever the gene copy is present, that is expressed. This is called pseudo dominance. Or if two copies of the same gene is present, one copy deleted or inactive, then the another copy automatically expressed. This is called pseudo dominance. So generally pseudodominance observed in haploid cases, or sometimes if two copies are present, one gene due to some mutation will become inactive, and opposite another gene or recessive gene express its character. So this is called pseudodominance. And third one is incomplete dominance. Incomplete dominant or partial dominance we studied in mirabilis cell form. I mean here the dominant and the recessive relation is not complete. Generally in dominance condition, the dominant gene 100% suppresses recessive gene, but incomplete cases, the dominant gene not 100% suppresses the recessive gene. It is less than 100, more than 50. So the dominant character, if any dominant copy or dominant gene potential is more than 50 and less than 100, then it is the incomplete dominant. You studied in mirabilis cell 4 o'clock p.m. So this is called incomplete dominance or partial dominance. Then epistasis. Epistasis means one gene is suppressed by another character coding gene allele. Then it is called epistasis. So the answer is epistasis. The process of suppressing one gene activity by another non-allelic gene is known as epistasis. For example, color coding gene is suppressed by height coding gene. Then it is called epistasis. So this is epistasis. Option is epistasis. Then next question is the genotypic ratio of monohybrid cross. If you take monohybrid cross, the phenotypic ratio is 3 is to 1. The genotypic ratio is 1 is to 2 is to 1. So here the genotypic ratio means first option is 1 is to 2 is to 1. This is right answer. Second option 1 is to 2. There is no such type of ratio. And third one also 2 is to 1. There is no such type of ratio. Last one is 1 is to 1. 1 is to 1 is the test cross ratio. Whenever test cross is given, monohybrid and dihybrid not like that. Whenever test cross is given, then the ratio is 1 is to 1. Whenever monohybrid cross is given, if they ask genotypic ratio, that is 1 is to 2 is to 1. If they ask phenotypic ratio, that is 3 is to 1. The 2 and 3 options are not at all exist. Fourth option exists. If you given test cross ratio, then you need to put 1 is to 1. If they given test cross, then we put 1 is to 1. Otherwise, monohybrid cross, what they asked is genotypic ratio. Genotypic ratio is 1 is to 2 is to 1, whereas phenotypic ratio is 3 is to 1. Take another question. The importance of satellite DNA is satellite DNA యొక్క ప్రాముఖ్యత ఏమిటి? You remember one thing, satellite DNA is all satellite DNA is non-coding in nature. So satellite DNA doesn't code any protein or RNA. So it is non-expression portion or non-coding in nature. So you remember this thing. All satellite DNA are non-express or doesn't encode any protein or RNA. So if you take first option, exhibit a high degree of polymorphism in population and the same degree of polymorphism in an individual that is heritable from parents to children. This is correct answer. So satellite DNA is highly repetitive sequence and it contains the entire sequence also. So that's why these are used in DNA fingerprinting technology. So satellite DNA doesn't code any proteins, any RNA. This is non-coding sequence, but it shows high polymorphism individual to individual. That's why they are used in DNA fingerprinting technology. So the first option is correct. If you take second option, the codes for protein of to be needed in cell cycle. As I said earlier, satellite DNA doesn't code any protein or RNA. It is non-coding sequence. So this is wrong answer. And third option is codes for enzymes to be needed in DNA replication. No. Satellite DNA doesn't code any protein or RNA. So it is non-coding in nature. Fourth option is all of the above. So first option is correct. It has high polymorphism and heritable from parents to progeny and so high degree of variations. So it is used as why this satellite DNA used in DNA fingerprinting. The next question is was discovered by Frederick Griffith. Griffith first identified DNA as the genetic material using bacteria by transformation experiment. Mouse experiment he performed using bacteria and perform transformation experiments. So Griffith identified DNA as the genetic material through transformation experiment. Central dogma was discovered by Watson and Crick. You know, transcription is the process of production of RNA from DNA. Any type of RNA producing from DNA is known as transcription. Whereas translation is the production of protein from mRNA. All types of RNA not produce protein. Only mRNA gives protein. So production of protein from RNA, that is mRNA, is known as translation. So mRNA converted into protein or mRNA decode into proteins is known as translation. Whereas transcription means any type of RNA, either tRNA, rRNA, mRNA, any other type of RNA coming from DNA. That process is known as transcription. Central dogma, DNA replication, DNA transcription, translation, all these things commonly called as central dogma. So Frederick Griffith identified transformation experiment. Through this transformation experiment, he used mouse and also streptococcus bacteria. So based on this, he identified DNA as the genetic materials. These are the questions, some of the questions of previous Yeah. Thanks for your listening.